So my name is Jennifer Kielczewski. We're at the National Institutes of Health, specifically in the National Eye Institute. We're in the Laboratory of Immunology. So we work on the eye, and specifically we work on uveitis, which is an inflammatory eye disease. Anytime you have inflammation inside the eye, specifically in the retina or any of those inside tissues, that's very bad and can lead to visual distortion, because people don't appreciate how important your vision is until you lose it. I first began interested in science. In elementary school, we went to NASA, which is in Greenbelt, Maryland, and I remember growing plants and flowers at a very early age and always very curious as to how things grow and why they grow. At an early age, I knew I wanted to do biology because I was always fascinated by the biological world, and I always asked my parents lots of questions, and they always told me, well, go and research it or look it up. So I began very early always looking up things and doing a research career. It's not a very boring type of job. It's a very exciting and enthusiastic career to have. So on a frequent basis, I do dissections as well as microscopy work, looking at ocular tissues and trying to figure out why things are inflamed and how to reduce inflammation in the eye in order to help people see better and to find new therapies and interventions to help prevent blindness. So here's the retina. The retina is commonly inflamed in uveitis. I'm pulling it away from the eye cup. And then I will cover slip it and flat mount it with a media, and then it'll be ready to be imaged. This is the retina that's been flat mounted. So now it is fluorescently labeled with a yellow dye that labels my ganglion cells. So I will go to the microscope and then image the retina flat mount and look for these specific retinal ganglion cells that are in the retina. And the retina has quite a depthness to it, so we use confocal microscopy to go through the retina, the different layers. So it's a very independent career. On a daily basis, I get to plan what I get to do. Every day I come to work and I get to run different assays and look at different parameters. I don't come in and look at the computer every day. I'm constantly in the lab doing something, working with other people, collaborations, going to meetings. So every day is a new adventure. So we're studying IRBP, which is interphotoreceptor retinoid binding protein. This is an important protein in photoreceptors. So I plan to look at this in another format, maybe such as Western blotting or some type of other protein analysis.
Just to conclude the observation from ELISA.
The lab is, it's a serious environment, but we have a lot of fun doing what we do. Poe. So there are lots of opportunities out there to explore science and to figure out whether or not you're a good fit for it. But I certainly encourage people to start exploring their choices in high school, even earlier, to figure out whether or not science is a good career for you.